Okay, my name is Dr. Robert J. Lefkowitz. I'm an investigator of the Howard Hughes Medical Institute and the James B. Duke Professor of Medicine and Professor of Biochemistry at Duke University Medical Center. I'm just thrilled to be getting this award at this stage of my career. I guess some would say perhaps my best work is behind me. I think that remains to be determined. I'm still quite active, but I see this as an award for really career achievement. And I've been at it a long time, probably about 40 years. So I would say I feel very fulfilled and satisfied and excited by the news of the prize.
Why did you focus your research on the transmembrane receptors in a time of general scientific skepticism about the role of single proteins in the signaling system?
When I was a very young investigator, I set out to study receptors because it seemed to me a field that was just ripe for breakthroughs. At the time, this would have been the early 1970s, the idea that there were receptors or specific molecules on cells with which drugs and hormones interacted was very controversial. Many, including some of the leading lights in pharmacology and medicine, did not specifically believe that such molecules existed. To me, based on several lines of evidence, it seemed clear that they did. And with what I would regard as sort of the arrogance of youth, I set out to sort of conquer the problem, never doubting for a moment that I would be successful. I've often looked back on those early days and thought to myself that if I knew then, just how difficult it would be, I'm not sure I would have had the gumption and the courage to go after it. But as a young guy in my early 30s, it seemed eminently doable. My choice of the particular receptors that I studied, though, had both a scientific and more of an emotional component to it. Scientifically, I wanted to study a receptor for which there were many drugs already available, because I realized that I would need to develop a variety of chemical and biochemical techniques which did not then exist in order to study the receptors. And the more structures of drugs that were available, the more molecules I would have to play with and to derivatize and to modify, etc. And not being a chemist myself, obviously the more molecules that were available, the better that would be. And this certainly would be true for the receptors for adrenaline, because there were many drugs already available, beta blockers and adrenaline itself and many other synthetic molecules. The other thing is I was a young, aspiring academic cardiologist, and so I wanted to study some receptors that had immediate and direct relevance to cardiovascular disease. And certainly the receptors for adrenaline certainly fulfilled that criterion as well. And so for both the scientific and more the emotional reasons, I chose the adrenergic receptors as the models for my studies.
The G protein coupled receptors can be found in every single cell of an organism. So is there any limit for the clinical application of your discoveries?
I think that there probably is no specific limit to the range of possibilities for drugs which target the G protein coupled receptor family. This is by far the largest family of receptors that exist in the human body. There are probably about 1,000 different receptors which share the structure which we discovered some 25 years ago, and which share many of the same properties. These include not only receptors for probably half the drugs in clinical use today, such as beta blockers, angiotensin receptor blockers, antihistamines, adrenaline, and on and on and on. But they also encode the receptors for many of our senses. We're able to perceive light because of a molecule called rhodopsin in the retina, which is a member of this receptor family. The senses of smell and taste and pain all are mediated by close relatives of the kinds of receptors that I study. So virtually every physiological process in the body is regulated, and hence I think there really is almost no limit at the present time to drugs which might be developed to target these receptors.
How do you feel about being honored with this important award at this moment of your career?
Well, it's a real delight for me to receive an award at any time, but particularly at this point in my career, when some perhaps might think my best work is behind me. I don't necessarily share that view. It's particularly exciting and motivating to me to receive this award for the work that I've done thus far. And I'm particularly honored to be in the company with Johan Massaguet, last year's winner, who is a good colleague of mine at the Howard Hughes Medical Institute. So all in all, I'd say it's a real good day for me.